Hello everyone. Today I want to talk about one of the most universally praised supplements in nutritional science, and I want to do it carefully, honestly, and with a level of nuance that most conversations about this particular topic never reach. Fish oil omega-3 fatty acids, EPA and DHA. The supplement with perhaps the strongest positive research profile in the entire category, genuinely anti-inflammatory, genuinely beneficial for cardiovascular function, brain health, and gut barrier integrity when taken appropriately. I am not here to tell you omega-3 south are harmful. The mechanism behind this gap is oxidation, and it is worth understanding in detail. Omega-3 fatty acids, EPA and DHA, are polyunsaturated fats. Polyunsaturated means they contain multiple double bonds in their carbon chain structure. Double bonds are chemically reactive sites. They are particularly susceptible to a process called lipid peroxidation, the reaction of the fatty acid with oxygen molecules that produces a cascade of degradation products. Lipid peroxides, malondialdehyde, quattro, idoxinanil, and a range of other aldehyde compounds, collectively called oxidized lipids, or Now here is the problem that makes this directly relevant to every person currently taking a fish oil supplement. Fish oil oxidizes rapidly. It begins oxidizing the moment the fish is processed, and it continues oxidizing throughout manufacturing, packaging, shipping, retail storage, and home storage. The rate of oxidation is determined by temperature, light exposure, oxygen contact, and the presence or absence of antioxidants. Room temperature storage, which is how most people store their fish oil, significantly accelerates oxidation compared to refrigeration. Transparent or translucent packaging accelerates it further. And the long supply chains between manufacturing and consumption provide months of opportunity for oxidation to progress before the product is ever opened. The second layer of the problem is what oxidized fish oil does specifically to the gut microbiome, and this is the dimension most people never encounter in standard omega-3 discussions. The gut microbiome is exquisitely sensitive to lipid oxidation products. The aldehydic compounds produced by fish oil oxidation, particularly for HNE and malondialdehyde, have been shown to directly damage the DNA of gut bacteria, alter bacterial membrane integrity, and selectively suppress the growth of beneficial bacterial species while promoting the proliferation of oxidative stress-tolerant genera that are associated with gut inflammation. According to research, dietary oxidized lipid exposure produces microbiome shifts within days that include measurable reductions in lactobacillus and bifidobacterium populations and increases in proteobacteria, a phylum whose members are consistently overrepresented in inflammatory gut conditions, including IBD and IBs. In one study, the microbiome changes produced by oxidized fish oil were distinct from and more adverse than those produced by saturated fat intake, previously considered one of the primary dietary drivers of dysbiosis. The oxidized lipid was producing more gut microbial disruption than a dietary input already known to be problematic. The third layer is the dose problem, which compounds the oxidation issue in a way that the research is beginning to take seriously. The anti-inflammatory effects of omega-3 fatty acids are dose-dependent. At moderate doses, roughly 1 to 2 grams of combined EPA and DHA daily, the anti-inflammatory mechanism operates through a competitive inhibition of the arachidonic acid pathway, reducing the production of pro-inflammatory eicosanoids. At higher doses, which have become increasingly common as the wellness culture around omega-3 south has intensified, a different set of mechanisms comes into play. Very high omega-3 intake suppresses immune function more broadly than intended, including aspects of immune surveillance that are important for gut integrity and pathogen defense. According to research, doses above 4 grams of combined EPA and DHA daily are associated with measurable immunosuppression in gut-associated lymphoid tissue, reduced production of certain antimicrobial peptides, and in animal models, increased susceptibility to gut pathogen colonization. The anti-inflammatory effect, that is beneficial at moderate doses, becomes an indiscriminate immune suppression at high doses. The fourth layer is the plant-based omega-3 false equivalence, which affects a different but equally large group of people. Many people who avoid fish-derived omega-3s south for dietary, ethical, or environmental reasons supplement with flaxseed oil, chia oil, or algae-derived ALA under the assumption that the body will convert it to EPA and DHA as needed. The conversion pathway exists. ALA is converted to EPA and then to DHA through elongase and desaturase enzyme activity. What the marketing does not prominently disclose is the conversion efficiency, which the research has consistently shown to be extremely low. The flaxseed oil is not delivering what the fish oil delivers. It is delivering ALA that will largely not be converted, while providing a substrate for oxidation that produces its own gut-relevant lipid peroxidation products, because ALA, as a polyunsaturated fat, oxidizes through the same mechanism as EPA and DHA. So what should you actually do? According to research, whole food marine omega-3 consumption consistently outperforms equivalent supplement doses in terms of bioavailability, tissue incorporation, and gut microbiome outcomes because the food matrix surrounding EPA and DHA in wild fatty fish includes astaxanthin and other antioxidants that protect against the lipid peroxidation that supplements are vulnerable to. The omega-3 is not the problem. The oxidized delivery vehicle is, and the solution is not more fish oil. It is better fish oil, or better yet, better fish. I hope this gave you something concrete to look at in your supplement cabinet tonight. Drop a comment and tell me whether you refrigerate your fish oil. The answer will probably surprise me. If this was useful, hit the like button and share it with someone whose fish oil has been sitting at room temperature for the past 6 months. And if you are new to NutriFlow, subscribe and hit the bell. See you in the next one.